Hello and welcome to today's webinar organised by NIST. My name is James Strachan, Deputy Editor of The Medicine Maker, and I'm delighted to be the moderator of today's online event, which is called The First Large-Scale Comparison of Glycoanalytical Techniques for Monoclonal Antibody Characterization in Industry and Academia. Our speaker today is Roisin O'Flaherty, Research Manager at NIST's Cell Technology Group. Roisin earned her BSc in Chemistry and PhD in Glycochemistry at Maynooth University in Ireland. She then travelled to Australia to do a research fellowship in glycochemistry at the Bio21 Institute, Melbourne University, before relocating back to Ireland to work as a process engineer at Intel. She subsequently joined NIST as a senior postdoctoral researcher, using her expertise in automation in the field of glycoscience. In 2019, she was appointed as a research manager in the Cell Technology Group at NIST, and she is responsible for the creation of some of the world's most advanced automated glycoanalytical technologies. She also has a strong focus on characterization of biotherapeutics and process development of monoclonal antibody cell lines. Roisin will be on hand to answer any questions you may have at the end of the presentation, but remember, you can submit questions anytime throughout the webinar by clicking on the Ask a Question box. Okay, we are now ready to start the presentation, so I'll hand over to Roisin. Roisin, over to you.
Thanks, Jim. So as Jim mentioned, this is a collaboration between NIST and the Medicine Maker, and today I'm going to talk globally about glycosylation in biopharmaceuticals. So the study I'll talk about today was initiated by the NIST, or the National Institute for Standards and Technology, which is based in the US. And it's a large-scale interlab study conducted across industry and academia to assess all the different types of glycoanalytical techniques for monoclonal antibody characterization. What I would like you to get, the reader, from this webinar in terms of your learning objectives is firstly to gain an insight into glycosylation and understand why it's so important. And secondly, I'd like you to observe some of the major trends in the glycoanalytical technologies that are out there and currently being used for monoclonal antibody characterization. And lastly, to gain an insight into some of the strengths and weaknesses of these glycoanalytical technologies that we will be talking about today. So if you can take home any of these, it'll give you a good introduction to glycosylation in biopharmaceuticals. Before I begin talking about the study, I'd like to just give a brief introduction to NIST. So NIST stands for the National Institute for Bioprocessing Research and Training, and we're based in Dublin in Ireland, and largely our remit is to do bioprocessing training and research in the area of biopharmaceutical manufacturing. So we support industry and academia in this area. For more information, go to www.nibert.ie. Broadly speaking, we have a really world-class training facility here at NIST, whereby we collaborate with some of the major biopharmaceutical companies across the globe. Here's just a spread of some of the training clients that we had in 2018. If we look at the research strategy that we have at NIST, we have four main pillars that we're interested in, all broadly related to the remit of bioprocessing. So we have interest in cell biology and engineering, bioanalytics, advanced manufacturing, and bioinformatics. And we collaborate with universities across the world and industry partners across the world, and we can achieve some of the funding either through private partnerships with industry or through some of the funding mechanisms like the European Union or Irish granting agencies. And then lastly in NIST, we have a contract bioanalytics team. And this might be quite important for you if you're interested in glycosylation as a service, because this is something our team at Contract Bioanalytics are experts in. So they can do glycan analysis, aggregate analysis, all types of biophysical analyses. And if you have any questions at all, just get in touch with us. So our contact details are www.nibert.ie. So to hone in on this study that we're interested in today, as I mentioned, we're going to be talking about a large interlab study that was conducted by the National Institute of Standards and Technologies. And it's important to point out that this webinar today is the perspective of me, one of the collaborators on the study, and doesn't necessarily reflect the views of NIST. So I just wanted to point that out. But before I begin talking about this large report, I think it's really important to gain a little bit of insight about Immunoglobulin G, or antibodies. So if we talk about biotherapeutics, 2/3 of all of the approved biotherapeutics in the world are glycoproteins. And glycoproteins are proteins which contain glycosylation. So that's 66% of biotherapeutics have glycosylation. So if you've never heard about glycosylation, or you have only a marginal interest in it, I would say be interested and try to gain more information about it. Of these glycoproteins, or biotherapeutics, which can include vaccines, fusion proteins, monoclonal antibodies, the vast majority of these are monoclonal antibody based. And of these monoclonal antibodies, a large cohort are based around the structure of IgG, or immunoglobulin G. So IgG, this is an antibody class found in our body, in our blood and extracellular fluid, and its main function is to protect against pathogens such as virus, bacteria, or fungi. Endogenous IgG is glycosylated. As you can see in this structure in front of you, there are 4 N-glycosylation sites in the Fab region, or the fragment antibody antigen binding region, and there are 2 N-glycosylation sites in the Fc region. Now, when we talk about monoclonal antibodies as opposed to endogenous IgG, generally speaking, the glycosylation is only found in the Fc region, so 2 sites of attachment for N-glycosylation. Generally speaking as well, when we are talking about glycosylation in this study, we're specifically talking about a particular class of glycosylation called N-glycosylation. And these N-glycans, if you look at the structure on the bottom left, are attached to an asparagine residue on an amino acid backbone, which is bound to another amino acid bound to serine and threonine on a protein backbone, So wherever you see the sequence of asparagine amino acid followed by serine or threonine, there's a potential for N-glycosylation sites. Now, the importance of characterization of glycosylation and some of the challenges are that, unlike the genome or the proteome, the glycome isn't template driven. In other words, we need enzymes and substrates to build up the glycan structures. So it makes the characterization a little bit challenging at times. But in general, for N-glycans, they follow somewhat of a structure template. You will always have, attached to the asparagine residue, 2 glucanac residues, which are then bound to 3 mannose residues, which you can see in the green. And these can then get decorated with further glucanac residues, more galactose residues, and sialic acid residues. And also in the context of IgG, we can get core fucose, which is bound to the innermost glucanac residue, which is that attached to the asparagine residue. So if you'd like more information about that, you can look at the review that we've published in Expert Review in Proteomics in 2018. So why is glycosylation important? Well, one of the really big reasons why glycosylation is important in the context of biotherapeutics is that glycosylation tightly regulates protein function. So the function of the protein, or in the case of monoclonal antibodies, the function will be altered based on the glycosylation of the protein. So just to give 2 examples, if you look at the terminal sialylation feature, those monoclonal antibodies that have higher levels of terminal sialylation, which are depicted by the stars, these will have a longer serum half-life in the body. And if you can imagine a monoclonal antibody or a therapeutic that has a longer serum half-life, it can lead to higher amounts of binding, which in theory could have higher immune responses. Similarly, with a high amount of terminal sialylation, we have decreases in ADCC activity. And ADCC activity is an effector function of the antibody, which is the Fc region, which interacts with FcR receptors on immune cells, and this initiates a protective functionality in our body. So if you wanted some of these features, you might want to have higher amounts of sialylation for your monoclonal antibody. Similarly, the absence of core fucose, which is that found on the innermost glucanac residue, increases ADCC activity. And this feature in particular is really harnessed by a lot of biopharmaceutical industries, where they've genetically engineered some of the cell lines to remove the corpuscles so that they can increase the ADCC activity. So this is one of the main reasons why glycosylation is termed a critical quality attribute by the regulators, is because it's so tightly bound to protein function. But the second reason it's really, really important, and probably a more important reason, is that it can influence the efficacy and safety of a biotherapeutic. So just to give one example, most of our monoclonal antibodies are produced in cell lines such as CHO cells, or Chinese hamster ovarian cells, or murine myeloma cells, mouse-based cells. And some of these cell lines have the ability to produce a sialic acid class called NGNA, or N-glycolyl-neuronimic acid. And this NGNA is not found in humans, and as such can be immunogenic. So even the smallest amount of the NGNA sialic acid that we can detect could be immunogenic to humans. So ideally, you'd like to be able to detect it in very low amounts and, if possible, minimize the amount of it. And similarly, if you talk about galactosylation as a feature, if you see an epitope of an alpha-gal bound to a beta-gal sugar in your IgG, or your monoclonal antibody, again, this epitope is known to be immunogenic. So it's something which you would want to characterize in very low amounts and to minimize. And then there are other glycan features which have other effects as well, but these are just two examples of why really glycosylation is so important to detect for biotherapeutics. So this brings us to the study that we're talking about today, it was published very luckily actually this month in October 2019 in Molecular and Cellular Proteomics, so it's hot off the shelf. As you can see, there are 168 collaborators involved in this project, 162 authors on this project, which stem from 73 labs worldwide across academic partners and industry partners. Everybody with the same remit to characterize a monoclonal antibody provided to us. So why did NIST decide to spearhead this huge study? They have a monoclonal antibody, a reference material 8671, which you can purchase on their website. And what they wanted to do was to use this monoclonal antibody to look specifically at the variation in glycosylation that can be observed, and to see how tight reproducibility between methods that you can observe. The second reason they want to look at it is that there are various different methods of glycoanalyses available throughout the world, and lots of different labs use different techniques. But there are very few examples of multi-lab studies, and particular with participation from industry partners. So taking these two together, they were very interested to spearhead this study. So the exact task was for the labs to identify and quantify N-glycans using a monoclonal antibody and a related monoclonal antibody. And the other goal of their study for NIST is to gain assigning consensus values for their NIST monoclonal antibody so that it can be used as a good reference with a lot of data behind it. So the strategy that NIST developed was, they did it in two stages. The first stage was they got participation from a few key labs, and gaining some of the insight that they got from this lab, they then initiated a much larger study which had advertisements, and anybody could join if they were interested in the study. The participants were provided with two samples, as I pointed out. So the NIST monoclonal antibody. Now, all the participants were told that it's an IgG1 kappa monoclonal antibody produced in an NS0 cell, so a mouse-derived cell. And we were also provided with another material, which is a modified material of the NIST monoclonal antibody, which has been stripped of some of the galactose residues. But we didn't know at the time as the participants that these were related to one another. We just knew that they were two monoclonal antibodies coming from an IgG1 cell line. And then the labs were asked to use any method of their choice to identify and quantify the end glycans in their samples. So this is just to give you an idea of the spread across the world of the participants in this study. As I mentioned, 76 labs participated, but what's particularly important about this study is the really high participation from industry. So as you can see, 43% of participants were from industry and 34% from university sectors. Even more importantly, in the industrial sector, you can see some of the service providers, the instrument providers, but almost 50% of those came from Biopharma directly. So that's either the R&D labs or some of the manufacturing sites. So this information contained within this report is really, really valuable. There are various different types of analysis that you can do to study the glycosylation of glycoproteins. So the first type of study that you can do is looking at intact protein analysis. And that's looking at the glycoprotein as it is without making any modifications or changes. And only 2% of participants used this type of analysis. The second type of analysis that you could do is you could use an enzyme such as trypsin or a cocktail of enzymes which will clip off smaller glycopeptide fragments from your glycoprotein. And as you can see, 21% of participants in this study used this type of analysis, of glycopeptide analysis. And then lastly, you could also use an enzyme such as pingase F, which is known to clip off N-glycans from the glycoprotein, and you just study the N-glycans in isolation. So 76, or the vast majority of participants in this lab, used this release N-glycan methodology. For this methodology, usually with glycans, they need to be visualized by LC or some type of technology, and fluorescent detection is one of the most popular choices. So for fluorescent detection, the pie chart on your right-hand side, you can see that 2AB or 2-aminobenzamide is the largest class that was used, followed by glycosylamine, which is at about 20% as well. And this incorporates some of the newer labels by some of the manufacturers, such as the RapiFluor labels or the Rapid labels. So the vast majority use that. I won't go into much detail of the pie charts on the bottom because I'll be describing here in these tables on the next slide. So of the 76 participants that conducted released N-glycan analysis, there's a breakdown in this table here. The number in parentheses are the number of labs that participated, and on the top of this table are those with the highest numbers, and on the bottom the least frequent participants used this technique. So I won't go through this painstaking table one by one, but you can look at it in the publication. But just to highlight the very top row, most participants, 20 of the participants, used 2AB labeling, so this 2-aminobenzamide labeling, as fluorophore. For their analytical technique, they used LC fluorescent detection, 15 of these participants, and some used it coupled to mass spectrometry. The chromatography was pretty specific to HILIC, which is a very common technique, or hydrophilic interaction chromatography, which is very ideal for glycans. But what you can see is, irrespective of the type of label that was used or the type of technology, for example, capillary electrophoresis is mentioned in there, and MALDI, which is quite common, and the identification techniques are quite varied, irrespective of what technology the users were using. So again, if you're looking at the 2AB label in the very top, mass spec is one form of identification which could be used, so the M over Z value. Additionally, standards were used by some labs. Other labs use retention time GU values. So this is a glucose unit value which is derived from comparing your retention time on your LC to a dextran lather, which can generate a GU value. And then other labs again use exoglycosidases or enzymes which can clip off sugars, and you can assign structures to them. If we look at quantification, peak area was used by most of the labs. So you can see there's a widespread even within the released N-glycan analysis, but the most commonly used would be using the traditional label of 2AB. If you look in more detail at the users that used glycopeptide analysis, in these cases, very few of the participants used any derivatization at all. So this is one potential advantage of this technique. And the analytical method of choice for glycopeptide analysis is LC-MS. The chromatography, because it's stemming from peptide analysis, uses reverse phase. So most of the participants, if you look at the top row, 17 of the participants use reverse phase. And in this case, the identification is quite different. They're using MSMS or MS directly for the identification of these compositions or glycan compositions. But again, quantification coming back to peak area. If you look at intact protein or protein fragments, very few participants used it. But again, these techniques were based around LC-MS or mass spectrometry as their major technique. So if you wanted to have a comparison of the different sectors and what technologies they used, this table is found in the supporting information, and it's quite informative in some ways. If you look at the values between the industry sectors, let's say, for example, and the university sectors, you can see that there's more of an even split between the participants that used a released N-glycan and glycopeptide analysis in the university sector, whereas for the industry sector, the users were more likely to use a released N-glycan analysis. And this probably comes back to the fact that traditionally speaking, a lot of biopharmaceutical companies wouldn't have the mass specs, whereas the university sectors would. And remember that the glycopeptide analysis was predominantly done using mass spec. The other interesting point from this table is if you look at the column that says glycosylamine, this had no participants at all from the university or other sectors, but a higher percentage of industry partners use this glycosylamine functionality as an alternative to 2AB. And again, these glycosylamine labels have increased detection in mass spectrometry and also have increased fluorescent detection. So there's major advantages to using them, but the downside is that they're more costly. And so probably in this case, the university sectors weren't using the more expensive labels. So just to give you a flavour of the type of analysis that we did at NIBRT, so we used an automated platform called a Hamilton Starlet. And remember, we were provided with 0.4 milligram of material of the monoclonal antibody and the reference material, which had galactose moiety stripped of it. But we were able to, just using 20 micrograms, and we could do it in replicates, in a 96-well plate format, we reduced, alkylated, and trypsinated the monoclonal antibodies. So if you imagine the IgG structure, what the reduction does is it breaks down the disulfide linkages which holds the structure together. Then the alkylation then alkylates these thiols so that they are fixed in position, and then trypsin will break the glycoprotein down into individual glycopeptide fragments. After that, pingase S will release some of the N-glycans, all of the N-glycans from the IgG structure, and then we can capture these N-glycans on a hydrazide bead using hydrazone chemistry, which will then allow for us to wash any interference salts off. You end cap the remaining positions on the beads, and then finally, when you're happy, you can release the glycans using mildly acidic conditions, dry them down, perform reductive amination using 2AB, label and clean up any excess label that you would have using solid phase extraction, resuspend it, and then perform the glycoprofiling using UPLC or ultra high performance liquid chromatography using separation based on Hillick chromatography. What we did was we generated GU values, which is comparing our LC profile to a Dextran standard, which generates these GU values. And these GU values can be fed into a database called Glycobase, which was developed in NIBRT, but which is now migrated into a database called GlycoStore, which has an array of GU values for distinct glycans. So that's one method of identification that we used. We also use mass spectrometry, LC-MS, And we also used a cocktail of exoglycosidase digestions, so enzymes which clip off certain structures, so that we could identify what structures were found in our sample. And just to give you an idea of how NIBRT fared in the study, if you look at these charts, on the x-axis is the unique glycan compositions, the number of unique glycan compositions that were identified in the study from all of the different participants, and on the y-axis the number of labs that identified these glycan compositions. So you can see for both of our samples we identified almost 40 unique glycan compositions in our study, and of these 40 glycan compositions, we were able to identify 99.9% of the samples that were in the monoclonal antibodies. So we were performed in the top 7% overall of all of the labs in terms of the highest number of unique glycan compositions. But just to point out the caveat that just because a lab identifies a high number of glycan compositions doesn't mean that they contain some of the structural information that other techniques might. And this is something I'll talk about a little bit later. So this is a really interesting breakdown maybe of some of the techniques. So the first example is the breakdown by method. And on the y-axis is the number of glycans reported. So you can see straight away that LCMS, if we look at these box plots, the line in the middle will indicate the median value of all of the participants, and the depth or width of the box plot will represent the number of labs that performed this analysis. And then the spread of the box plot indicates 50% of the participants. And so you can see that the highest value was obtained using LCMS. Highest number of unique compositions were obtained using LCMS-based techniques. If you split it by analyte, the largest class would be those that used glycopeptide analysis instead of released N-glycan analysis. And similarly, if you broke it down by sector, you can see that the university participants reported the highest number of glycans compared to, let's say, the industry partners. Now, you could make an argument here that the industry partners were doing a lower risk strategy, and that they wouldn't report any glycans that they weren't absolutely certain of, and it's possible that some of the university partners were putting compositions that they were less certain of. So I take this information quite lightly, but just to highlight that you'll get the highest number at present from LCMS, but not necessarily the highest level of depth of information, so keep that in mind. If you want to think about the depth of information or the highest proportion of isomers that were identified were actually identified with those technologies which incorporated exoglycosidase digestions. And the reason for that is that mass spectrometry can't differentiate isomers because they have the same M/Z values. So in order to identify some of these isomers and some of the structural motifs, we need to include some other technologies such as exoglycosidase digestions, or maybe there are some other technologies in the future like ion mobility which could play a huge role to gain more structural insight. Also in this publication there is a series of tables which provide all of the different consensus values that were reported in this study. I'm only showing you the very beginning of the table, but for example, the most highly identified glycan in this study was G1F, and this G1F contains one fucose residue and one galactose residue. Every single lab, 103 reports, identified correctly this G1F structure, and the median value is 39% approximately of the sample contained this structure G1F. What's interesting about this is for these very abundant glycans at the top, the CV values of the coefficient of variance was quite small. So for this top hit, it was 6% of a CV. Because if you remember, the participants used a huge array of different techniques and there were variations along the way. So to have this good agreement of this percentage area or quantification of this glycan is pretty good. Now you will notice as you look down through the table that these values get larger and larger as the less consensus exists for the glycans. Also in this paper, just to give you a highlight, if you're interested in measuring glycosylation features, there is a breakdown provided in this table. This might be important if, for example, you were creating a new monoclonal antibody and you wanted to make sure that you were characterizing low amounts of the immunogenic epitopes, such as the NGNA or N-glycolyluronic acid sialylation, or, for example, that alpha-galactosylation that I mentioned earlier. You might want to compare this monoclonal antibody to see what values you're getting relative to the community, and then use your own new monoclonal antibody. So this could be quite important. Also in this study, they provide these box plots. On the top, you can see the sample, which is the NIST monoclonal antibody, and each box plot will represent a unique composition of that was identified in the study. Again, as earlier stated, the line in the middle will indicate the median. The spread or the width of the box plot will indicate the number of participants that reported this glycan, and the spread on the y-axis is the deviation between methods. So, the graph in the middle will show the ratio of the monoclonal antibody to the reference material. And as I mentioned, the reference material is only different in terms of the galactose residues. So you can see all those compositions in the center weren't altered by the treatment of the galactosidase, which makes sense. But overall, you can see from these box plots, there's good agreement amongst the different labs. On the right-hand side, the target plot just shows you the agreement between the labs as well. On the x-axis is variation in the standard deviation, and on the y-axis is the spread amongst the mean values that were reported. So each point on this target plot is a summary data based on an algorithm of every lab. Their data was fed into this, and it gave one distinct value from each lab. And the circles represent the standard deviations. So if you imagine all them points in green, they're all within 2 standard deviations of each other. Those that are in yellow are 3 standard deviations, and those outside of that would be outside of that. So you can see that in general there is very good agreement amongst the participants using the methods. And if you broke this down by what analysis type that was used, on the top left, you can see there's no clear advantages to using released glycan or glycopeptide or other technologies. On the right-hand side, you can see that, for example, mass spectrometry isn't outperforming fluorescent detection or vice versa. You can see that the industry partners aren't outperforming the academic partners. But what you can notice in the target plot on the right-hand side, bottom right, is that those participants that had a higher number of replicates, those that are depicted in green, had less deviation. So this kind of comes back to those users that had better lab design and practice with a higher number of replicates, got better consensus values for the N-glycans in this study. Also in this paper, there are some examples, and there's really a few tables here which outline some of the pros and cons of different glycoanalytical techniques. There are various techniques available, as I mentioned, capillary electrophoresis, there's UPLC, there's MALDI, there's LC-MS-based techniques. And each one of these techniques has pros and cons. So for example, that top hit was a lab that used sialic acid derivatization. And one of the strengths of this technology was that they were able to identify and differentiate 2 isomers of the G1S structure, which other technologies weren't able to identify. But one of the weaknesses was that there was an overlap of 2 particular glycans in this method. And if you imagine a HPLC chromatogram and you're integrating a peak and there's more than one glycan in that peak, it's very difficult to quantify exactly the abundance of one of the 2 species or one of the 3 species. So I would say in general, and this is just my personal take home, but in general, UPLC or HPLC based methods where there might be a weakness is in the overlapping of some glycans within a peak. and therefore mass spectrometry would be better in some techniques when you think about that. But one of the downsides of using mass spectrometry as a standalone is that it can't differentiate between isomers. So glycopeptide analysis generally uses oxonium ions to identify the N-glycans that are present, and they can't always get the structural information. So for the highest level of structural information, you need to use a combination of technologies, such as using exoglycosidase digestions or databases, and some of these tricks like derivatization of the sialic acid, but also to gain the highest level of unique glycan compositions, mass spectrometry is definitely valuable. So in conclusion, we generated a list of consensus values for the NIST monoclonal antibody, which, as I mentioned, is available on the website. This study provides a baseline for the current state of art of glycosylation, so it gives us an idea of what techniques participants are using out there. And in addition to this, it really highlights that there is a clear need for harmonization amongst techniques. So if you remember that table I was showing you with all the different types of identifications that are out there, I think there's a need both for regulators and for us as glycoscientists to try come up with a more standardized approach, because at present there's so many different types of analysis that you can do. And then lastly, this really showcases a universal basis to compare your newer glycoanalytical techniques to those that are derived from, let's say, the community. So it gives you some confidence in the technique that you have. And I just want to leave you on this final note where, in my opinion, as Thomas Edison stated, there is a way to do it better, find it. And I really think this is shines through in this study that at present, glycoscience is a relatively young field and we have a lot to do to advance it forward, but we're really making huge strides. And personally, I think one of the ways that we can do this is to merge technologies together, so to potentially use some of the strengths of some technologies and merge them with some of the other technologies, and this will give us the highest level of depth of information and the highest number of compositions, so to get really low abundances of certain glycan epitopes, which, as we know, might link back to safety and efficacy. I'd just like to thank, so at the time of the study, I was a postdoctoral researcher working in the laboratory of Professor Pauline Rudd. She's on the bottom right-hand picture of the main picture, and also Dr. Radka Saldova was involved in this study. I've since moved, and I'm the research manager in the cell technology group at NIBRT, which is spearheaded by Professor Mike Butler on the right-hand side of the screen. And I just want to say a particular thanks to Dr. Lorna DeLeo, who initiated this study from NIST, and she has since moved to Agilent, but at the time she was the person collating all this information from all of the different labs, and I can imagine how painstaking that was. And then lastly, if you're interested in collaboration in terms of biotherapeutic characterization or manipulation, please feel free to contact me. My email address is provided on the screen, and my Twitter account @IrishGlycoGirl.